Well, immersing with the effect of temperature on microbial growth, because temperature is the most influential factor which affects on microbial growth. And we know that on the basis of temperature, microorganisms has been divided into four to five groups. Those are sacrophiles, mesophiles, thermophiles, hyperthermophiles, and thermophiles. But how microorganisms are getting adaptation in extreme temperature, suppose at high temperature or at low temperature, we have to see it in detail. Suppose we are looking microbial life in cold. When temperature is very low, below than 10 degrees C or in minus degrees C, so microbes are there, but how they are surviving over there, we have to see this thing. Extremophiles organisms that grow under very hot or very cold conditions are called extremophiles. Extreme means very extreme environment. Okay, philes means lovers. Sacrophiles organisms with cold temperature optima. So sacrophiles are those organisms which can survive, which can grow at cold temperature. Their optimal growth is on very cold temperature. So those are called sacrophiles, inhabit permanently cold environment. Just they can live in cold environment. If someone will change or transfer them into another place, so they cannot survive, they cannot grow over there. And sacrotolerant are those organisms which can grow at low temperature, but optimal temperature is up to 20 degrees C to 40 degrees C. So such type of microorganisms which have high tolerance range are more widely distributed in nature than sacrophiles. That sacrophiles are limited. They can live only in cold temperature, but sacrotolerant have ability to grow at a varying range of temperature. Their optimal temperature, they can grow at 0 degrees C, but their optimal temperature is 20 to 40 degrees C. So this range of temperature ranges in most of the habitats of this globe. So such microorganisms are widely distributed in nature. Molecular adaptation to psychrophilia. How microbes get adaptation to cold environment. So we can see over here, microbes can produce such enzymes that function optimally in the cold temperature. So microbes have ability to produce such enzymes which have ability to function at cold temperature. Features that may provide more flexibility. Features that may provide more flexibility, that they can work at cold temperature. So microbes which can grow at very cold temperature have more alpha helix than beta sheets. This is adaptation. More polar and less hydrophobic amino acids they have. More polar and less hydrophobic amino acids they have in their cytoplasmic membrane. More alpha helices than beta sheets are there. More polar and less hydrophobic amino acids are there in their cytoplasmic membrane. Fewer weak bonds are present. Decreased interaction between protein domains. So these are the features which are present in microorganisms which can live at low temperature, at cold temperature. In their cytoplasmic membrane, they have ability to produce certain enzymes which can work at very low temperature optimally. Their proteins have more alpha helix sheets than beta sheets. This is one difference. More polar and less hydrophobic amino acids are present. Fewer weak bonds are present. Decreased interaction between protein domains. So microbial life in the cold, molecular adaptation to psychrophilia, transport process function optimally at low temperature. So this is another function. Transport proteins which are present in cytoplasmic membrane. We have studied in detail in our second chapter that transport membrane, transport proteins are present in cytoplasmic membrane, which have ability to transport, which have a function of selective permeability, just allow to certain molecules uh, to come inside or go outside. So these are working at low temperature very well. Therefore, microbes can survive at low temperature very well. And their modified cytoplasmic membrane, also have changed. So high unsaturated fatty acid content is present in cytoplasmic membrane. This is most important feature. Cytoplasmic membrane is playing more important functions. So cytoplasmic membrane have high unsaturated fatty acid content, which is present in other eukaryotic animals also, which are living at very low temperature. So here you can see this is a thermal vent. This is a hot spring. From here sample has been collected and have seen that microbial populations are present. Only rods are present. Mostly gram-positive rods are present in this thermal vent. And in cold environment, if you will collect samples, we will find the same saccharophilic microorganisms. So microbial life at high temperature. So we have studied that how microbes can survive, grow, and function at low temperature. We have seen some basic changes. What are some basic changes? These basic changes are enzymes, which are present in cytoplasmic region of microorganism and produced by cytoplasmic membrane, having more flexibility, more alpha helix than beta sheets in proteins, more polar and less hydrophobic amino acids are present in different structures, fewer weak bonds, decreased interaction between protein domains, and the transport process functions optimally at low temperature, modified cytoplasmic membrane having high unsaturated fatty acid content. Okay, so how microbes can survive at high temperature? This is our next step. Studies of thermal habitat have revealed that prokaryotes are able to grow at higher temperature than eukaryotes. So it is one thing clearly that microorganisms have more ability to survive at high temperature or cold temperature compared to eukaryotic organisms. Eukaryotic organisms have a range. If conditions or factors will increase beyond those limits, they will be unable to survive. But microbes have ability to survive beyond it is low range. Suppose prokaryotes are able to grow at higher temperature than eukaryotes. Organisms with the highest temperature optima are archaea, microorganisms which can grow at thermal vents at high temperatures are archaea. Non-phototrophic organisms can grow at higher temperature than phototrophic organisms. Non-phototrophic we should consider, we should understand this point that non-phototrophic organisms can grow at higher temperature than phototrophic organisms. Molecular adaptation to thermophilia, that how microbes have little bit change compared to mesophilic and saccharophilic microorganisms to grow at high temperature. Number one, again, enzyme and proteins function optimally at high temperature. Features that provide thermal stability. That microbes, those microorganisms which are living at extreme high temperatures have ability to produce certain enzymes which can optimally function at high temperature. This is the one adaptation, and it is genetically controlled. Number two, critical amino acid substitutions in a few locations provide more heat tolerant folds. So this is one thing. Critical amino acid substitutions are present in a few locations provide more heat stability. That in some places, instead of amino acids, some unusual substances or substitutions are present which are providing more stability to amino acids in structural and functional proteins. An increased number of ionic bond between basic and acidic amino acids. This is second adaptation. An increased number of ionic bonds between basic and acidic amino acids resist unfolding in the aqueous cytoplasm, which have resistance for unfolding in the aqueous cytoplasm. Number one, critical amino acids are present where some substitutions are present instead of normal amino acids. Number two, increased ionic bonds are present between acidic and basic amino acids, which are making it resistant to unfold in aqueous cytoplasm. Number three, production of solutes, suppose diacetyl phosphate, diglycerol phosphate help stabilize proteins. Microbes which are living at high temperature, they have ability for the production of certain solutes, glycerol phosphates and diacetyl phosphates, which are stabilizing proteins. So molecular life at high temperature, again, molecular adaptation to at high temperature, modification in cytoplasmic membrane to ensure heat stability. There are some modifications which are present in cytoplasmic membrane of those microorganisms which can grow at high temperature. What is that? Bacteria have lipid rich in saturated fatty acids, and archaea have lipid monolayer rather than bilayers. These are two modifications in those bacteria which are living at high temperature. Bacteria have lipid rich in saturated fatty acids, and archaea which are living at most higher temperature have lipid monolayer rather than bilayer in cytoplasmic membrane. If you have seen cytoplasmic membrane in our previous class, that is bilayer of this fatty acids, but in archaea there is one layer, monolayer. Hyperthermophiles produce enzymes widely used in industrial microbiology. Microorganisms which can grow at high temperature are very important for industrial purpose, because when they are producing enzymes, they can be at high temperature, they can be used in industrial process, because industrial process have uh, high temperatures. Because in fermentation reactions we are using high temperatures. So microorganisms which can produce enzymes, industrially important enzymes at higher temperature, they are very important for commercial use, because in commercial production of enzymes it needs high temperature. If enzyme is stable in high temperature, so that is most beneficial. Example, take polymerases. This is used in polymerase chain reaction. The stack polymerase is used to automate the repetitive steps in polymerase chain reaction PCR technique. So this enzyme is produced by microorganisms, and this enzyme is stable at high temperature. Therefore, it is very important. Other environmental factors affecting the microbial growth: acidic and alkalinity, osmotic effect on microbial growth, oxygen and microorganisms, toxic forms of oxygen. So various environmental factors. We have studied high temperature, adaptation of microorganisms in high temperature, adaptation of microorganisms in low temperature, and we have also to discuss other environmental factors affecting the microbial growth in environment. Those are acidity and alkalinity, means pH of environment is affecting microbial growth, and osmotic pressure also affecting the microbial growth, oxygen also affecting the microbial growth and having important role in microbial population proliferation. Toxic forms of oxygen also playing a vital role in environment. So we are talking about here the acidity and alkalinity, how acidity and alkalinity affecting the microbial growth. So if you will see, the pH of an environment greatly affects microbial growth. We should be clear. Some organisms have evolved to grow best at low temperature. Now, some microorganisms have, and some organisms have evolved to grow best at low or high pH. So, this thing, acidity and alkalinity, the pH of an environment greatly affects the microbial growth. Suppose if pH is near to neutral, most of the microorganisms will grow over there. If pH is acidic, only acidic microorganisms will grow. If acidic pH is alkaline, only alkaline microorganisms will grow. Some microorganisms have evolved to grow best at low or high pH, but most organisms grow best between pH 6 to 8. So up to this, we are keeping a stop over here, and from here we will study in next class. You must have to go through our textbook which has been recommended in your course content, and Prescott soft copy also has been provided to you, so you can debate, you can read it from there, you can ask the questions online, or you can discuss it with your colleagues and friends. Thank you. Have a nice time, please.